Hello, good morning. Myself, I am BSR Botany lecturer. Today, our topic is photosynthesis, dark reaction. In the dark reaction, already C3 pathway completed. Now the C4 pathway and also CAM pathway. First C4 pathway. C4 pathway first reported by Hatch and Slack. Especially some of the plants which are growing in tropical and subtropical regions of the world, and there the temperature is about 35 degrees centigrade to 40 degrees. In that plant, example sugarcane, the first stable compound is a 4 carbon compound. In C3 plant, the first stable compound is 3-PGA. There the carbon, 3 carbons are present. Hence, the pathway is called C3 pathway. Whereas here 4 carbon compound, oxaloacetic acid. So that is why this cycle is called C4 pathway. So the process here in C4 plants, two types of cells are there: mesophyll cells and bundle sheath cells. Especially here, Cron's anatomy is present. What is the Cron's anatomy? The vascular bundle surrounded by bundle sheath cells, which appears like a garland, that is called crans. That Cron's anatomy is present in the bundle sheath cells. Anyway, in the C4 pathway, mesophyll cells and bundle sheath cells, two cells are involving. In the coordination of mesophyll cells and bundle sheath cells, only C4 pathway can complete. So here the process. The first phosphoenol pyruvic acid, that is the 3 carbon compound, binds with the carbon dioxide. Carbon dioxide is available in the form of bicarbonate ion. So phosphoenol pyruvic acid condensed with the carbon dioxide to form 3 carbon plus 1 carbon, 4 carbon compound, oxaloacetic acid is formed in presence of phosphoenol carboxylase, PEP phosphoenol pyruvic carboxylase. So anyway, oxaloacetic acid is formed, so 4 carbon compound. Now the oxaloacetic acid moves from cytosol of the mesophyll cells to chloroplast of the mesophyll cell. That movement is intracellular transportation, intra, not inter. Now their 4 carbon oxaloacetic acid reduced to malic acid. For that reduction, NADPH2 is required. That is the light product. So oxaloacetic acid reduced to malic acid in presence of malic dehydrogenase. Now this malic acid moves from mesophyll cells to bundle sheath cells. The transportation is intercellular transportation. So chloroplast of mesophyll cells to chloroplast of bundle sheath cells. There the malic acid undergo oxidative decarboxylation. Oxidation means NAD converted into NADPH2. Here one is utilized, one is synthesized. So balance is over. Now oxidative decarboxylation, removal of carbon dioxide is also takes place. 4 carbon compound converted into 3 carbon compound. Now this carbon dioxide entered into the PCR cycle in the bundle sheath cells. In the bundle sheath cells, the carbon dioxide entered into the PCR cycle. Especially in the bundle sheath cells, chloroplast is agranal chloroplast. Here agranal, no grana. It means only dark reaction. Whereas here granal chloroplast, here grana is present. That is called chloroplast dimorphism can be seen. Chloroplast dimorphism. So agranal chloroplast, their PCR cycle takes place. Here important point. In these C4 plants, especially the C3 pathway is not depending on atmospheric carbon dioxide. This carbon dioxide is released by the reactions. So that is why in the C4 plants, no photorespiration because rubisco never act as oxygenase. Rubisco always act as carboxylase because it is not depending on atmospheric carbon dioxide. It is depending on the carbon dioxide is formed as a result of reactions. So that is why the C4 pathway more effective than C3 pathway. So then malic acid converted into pyruvic acid because of oxidative decarboxylation. Now the pyruvic acid again moves from chloroplast of bundle sheath cells to chloroplast of mesophyll cells. There the pyruvic acid undergo phosphorylation. Especially for that, two ATPs are required in presence of pyruvate dikinase. So pyruvic acid converted into PEP. So again the PEP involved in process. PEP carbon dioxide oxaloacetic acid. So anyway, here important thing in the C3 pathway for synthesis of 1-glucose, how many ATPs are required? 18 ATPs are required. How many NADPH2s are required? 12 NADPH2s are required. So how many ATPs and NADPH2s are required for C4 pathway? That is the important thing. So here, for synthesis of 1-glucose, how many carbon dioxides are required? 6 carbon dioxide. It is very clear. Just you observe, for 1 carbon dioxide, how many ATPs are utilizing? 2 ATPs. Like that, for 6 carbon dioxide, how many ATPs are required? 6 into 2, 12. So 12 additional ATPs are required. Generally, for synthesis of 1-glucose, 18 ATP, 12 NADPH2. Along with that, additional 12 ATPs are required. Like that, in the C4 pathway, how many ATPs are required? 30 ATPs are required and 12 NADPH2s are required. So this is the C4 pathway. Then again you go for CAM pathway, crassulacean acid metabolism. The crassulacean family members like bryophyllum, their scotoactive is present. So scotoactive means stomata open during night time, closed during daytime. That is the specialty of crassulacean family members. So in the crassulacean family members, during the night time, so the phosphoenol pyruvic acid binds with the carbon dioxide during the night time because stomata open during night times only. So CO2 enter during night time. So phosphoenol pyruvic acid, that is the 3-carbon compound, binds with the carbon dioxide to form 4-carbon compound. So phosphoenol pyruvic carboxylase peptidase enzyme is required. Here also the first stable compound is OAA, oxaloacetic acid, that is formed during night time. Now OAA reduced to malic acid. For that reduction, NADPH2 is required. NADPH2 converted into NADP in presence of malic dehydrogenase. Malic dehydrogenase. Now this malic acid stores in the vacuole. That is storing in the vacuole. So they stores in the vacuole up to daytime. But during the daytime, now the malic acid undergo oxidative decarboxylation. Oxidative decarboxylation means oxidation, NADP converted into NADPH2. Here utilize their synthesis. Decarboxylation, removal of carbon dioxide. Again this carbon dioxide entered into the PCR cycle. Okay. So here malic acid 4-carbon compound, when oxidative decarboxylation takes place, 3-carbon pyruvic acid is formed. Same process, pyruvic acid undergo phosphorylation. So phosphorylation, for that phosphorylation, how many ATPs are required? 2 ATP. In presence of pyruvic dikinase. Finally, 3-carbon phosphoenol pyruvic acid is formed. Again, during the night time, same. Phosphoenol pyruvic acid condensed with the carbon dioxide to form oxaloacetic acid. Same cyclic process takes place. Here also same, how many ATPs are required? 30 ATPs are required and 12 NADPH2s are required. 12 NADPH2s are required for synthesis of 1-glucose. But this process takes place during night and daytime. So this is C4 pathway and CAM pathway. Thank you. Thank you to all.